Good morning and welcome. We're going to be starting the public webinar in about 1 minute, but you are at the webinar, the ICER webinar on AMX and oral Aderavone for amyotrophic lateral sclerosis. I'm Steve Pearson. Please stay tuned. We'll be starting in approximately 1 minute.
Good morning, everyone. My name is Reem Mustafa. I'm a professor of medicine at the University of Kansas, and I will be chairing this meeting today. Welcome to this session. Like Steve just said, we will be discussing 2 different medications today for ALS, and I would like to start by welcoming you all. At the beginning of every CEPAC meeting, we want to ensure that all speakers disclose any financial relationships with industry. Every member of our CEPAC panel who is present today has met the ICER conflict of interest policy. I would ask each of the CEPAC members to introduce themselves, their work, and note whether they have any updates to their declarations. And I'll start by stating that I do not have updates to my declarations. Dr. Armbach, if you want to introduce yourself.
Hi, my name is Eric Armbrecht. I'm a professor at St. Louis University in the School of Medicine in the College of Public Health. I have no disclosures to add today. Thank you.
Thank you. Dr. Balk.
Hi, I'm Alan Balch. I'm the CEO of the National Patient Advocate Foundation, and I have no updates to my disclosures. Thank you.
Thanks. Dr. Bora.
Yeah, hi. I am a professor of health services research at the Mayo Clinic College of Medicine. I have no updates to the disclosures that I already made earlier.
Great, thank you. Dr. Carroll.
I'm Aaron Carroll. I'm a professor of pediatrics and chief health officer at Indiana University, and I have no updates to my conflicts of interest.
Thank you. Dr. Casey.
Good morning, Don Casey, Associate Professor of Internal Medicine at Rush in Chicago, also affiliate faculty at the Jefferson College of Population Health in Philadelphia. No disclosures or updates.
Thank you. Dr. Kerfman.
Good morning, I'm Greg Kerfman. I am a cardiologist by training, a deputy editor at JAMA, the Journal of the American Medical Association in Chicago, and on the faculty at Harvard Medical School. I have no conflicts to disclose.
Thank you. Ms. Dave.
Hi, my name is Sneha. I'm the Executive Director of Generation Patient, a nonprofit focused on adolescents and young adults with chronic and rare conditions, and I have no disclosures to report.
Thank you. Dr. Dusetzina.
Good morning, Stacy Dusetzina. I'm an Associate Professor of Health Policy and an Ingram Associate Professor of Cancer Research at Vanderbilt University Medical Center, and I have no updates to my disclosures.
Welcome, thank you. Ms. Brown.
Good morning. My name is Angela Brown. I'm CEO for the St. Louis Regional Health Commission, and I have no disclosures or updates.
Great, thank you. Ms. Guidon.
Good morning. My name is Heather Guidon. I'm a board certified patient advocate and the program director of the Center for Endometriosis Care. I have no updates to my disclosures and no conflicts of interest.
Excellent. Welcome. And next on my list, I seem to have lost my list for a second here. Dr. Johnson.
Good morning. I'm Jill Johnson. I'm a pharmacist and professor of pharmacy practice at the University of Arkansas for Medical Sciences College of Pharmacy. I have no updates to my conflict of interest statement. Thank you.
Welcome. Thank you. Dr. Martin.
Good morning. I'm Brad Martin, professor in the Division of Pharmaceutical Evaluation and Policy. I also work at the University of Arkansas for Medical Sciences and have no updates to my conflicts.
Welcome, thank you. Dr. McBride.
Good morning, I'm Timothy McBride. I'm a professor at the Brown School at Washington University, and I have no updates to my conflicts.
Great, and Dr. Will?
Hi, I'm Timothy Wilt. I'm a professor of medicine, general medicine, and public health, health policy and management at the University of Minnesota, a core investigator and staff physician at the Minneapolis VA. I have no conflicts and no updates. Thanks.
Excellent. Thank you all. And I would like now to introduce our patient and caregiver experts. Ms. Colette?
Hi, I'm Kathy Collett in Indianapolis, Indiana. Was one of the caregivers to my mom who died from ALS 25 years ago, and I made disclosures to ICER that I think should be up to date. Thank you.
Thank you very much, and thanks for joining us. Dr. Shamaskin?
I'm Joel Shamaskin. I'm a retired professor emeritus of medicine from the University of Rochester, and I have no updates to my disclosures.
Thank you very much, and thanks for joining us. And we also have our clinical experts, Dr. Bidlack.
Hello, everyone. My name is Dr. Rick Bedlack. I'm a professor of neurology and director of the ALS clinic here at Duke University. No updates to my disclosures.
Thank you. And Dr. Lewis.
Good morning, everybody. Aaron Lewis. I'm a neurologist and neuromuscular specialist at Kaiser San Francisco, and I run the ALS clinic and direct the clinics for ALS across Northern California for Kaiser.
Great, and thanks all for joining us. I'm going to request that anyone who speaks, including in the policy roundtable, to actually announce any of their financial conflict of interests. And with that, I would like to introduce Dr. Steve Pearson, President of ICER, who will take us over some introductory slides. Steve, to you.
Thank you, Reem. And again, good morning to everybody, and thank you in advance for the participation of everybody over the 8 months, but in particular today, the patient experts and the clinical experts who are joining us for this discussion. I want to do just a little bit of level setting around kind of why we're here today, the purpose and some of the methods that are kind of supporting this meeting today. And I want to start with a comment from a person living with ALS. The initial symptoms are frustrating and worrisome, regardless of how fast or slow they come on. You need to find support, how to prepare your house and life for this. You need moral and psychological support. You have family who are often just as upset, if not more upset than you are. A major preoccupation is trying to figure out the extended period of time where you need expensive home care. It makes you wonder if you're going to bankrupt your family. Now, this is obviously one person's story, and like almost all conditions, the experience of living with ALS, of the individual, of the family, can be very different. But it obviously is a condition that we have become more familiar with through the activity of the patient advocacy community and others. And we know how deeply it affects both patients and their families. So that is the primary reason that we are here today, is to focus on that. And because we have new treatments, relatively new, and those that are still in the pipeline and under evaluation at the FDA. And the reason we're here in part is because things start to happen the day that the FDA approves a new treatment. Now, again, we'll talk about AMX and we'll talk about Aderavone as individual treatments, but in general, in this kind of situation, when the FDA approves a new treatment, what happens is a sense of celebration. There's a sense that the years of investment, of risk-taking by companies, by patients and the trials, by scientists in the labs, all of that works together to provide a new opportunity to help patients and their families. And so there is a justifiable sense of celebration and an eagerness to use new treatments to help patients. We all know that at that same time, the day that the FDA approves it, there are going to be questions. There are still going to be questions about the evidence. it's going to be relatively new. And so people are going to be wondering, what are the risks and benefits for different types of patients? How do they balance out? How do we know for whom it's best intended? And part of that is, how do they fit into the evolving landscape? There might be a new treatment coming next week that will be even better, or there could be certain reasons to wait or not wait. And so we have to think about all of this at the time that a new treatment becomes available. And then there are also going to be questions about costs. These are costs to the patients themselves, to the health system, and to the government, because in our country, at launch, at the government level, there's still no organized federal process for price negotiation. The new law that was just passed will negotiate many years downstream a limited number of drugs. But we're still facing a general ecosystem in which that early launch price is unconstrained, and it's up to the manufacturer to set the price in the context of their thinking about the environment. So those questions become very real, even on day one. And we're always thinking about whether we're going to be learning lessons or providing lessons to help us in the future, to do better by patients, to help create a good, innovative ecosystem that is also affordable for everybody. So we're here for all of these reasons. And part of it is that we're going to be focusing on ALS and people with ALS. But part of the reason we're here today is because of the broader aspects of rising healthcare costs and its impact on others. We'll talk a fair amount about this, but in our country, we still have a real issue, every country does, with trade-offs and with problems related to the cost of healthcare. There was a very well done survey and an article about it in NPR, but it was from Kaiser Health News. Sorry, that was the outlet, but it was also reported in NPR. The Kaiser Family Foundation did a survey of Americans trying to explore deeper issues related to medical debt. And what they found was pretty shocking. They found that over 100 million Americans, by extrapolation from this survey, with a pretty good level of precision, over 100 million Americans are estimated to be currently in medical debt. A lot of that is being carried on credit cards. It's not all visible through being in receivership, et cetera. But this is a lot of people, and they ran through a number of individual stories. The folks on the left, Marcus and Allison Ward of Chicago, they fell into debt when they had premature delivery of their twins. They had to change jobs, move to live with family, and they're currently still about $80,000 in medical debt. The couple in the middle, that's Samantha and Ariane Buck from Arizona, he had an intestinal infection and kind of didn't have enough money to afford health insurance and was running bare in terms of not having insurance, thought he was healthy, had this infection, and now they are in debt over $50,000, and they're preparing medical bankruptcy. And the woman on the right is Sherry Foy from West Virginia. She was at the tail end of an employer kind of sponsored insurance. She thought she had good insurance, but it had a cap on it. And when that was exceeded, she ended up being more than $600,000 in medical debt and also had to file for personal bankruptcy. Now, these are just individual stories, but I think it's helpful to remember that these are individuals in our community, 100 million Americans, not all of them this severely affected. But of those people who responded that they were in medical debt, just a couple of other facts. Of those, 63% said that they had to cut their spending on food, clothing, or other basics. 48% said they had used up all or most of their savings. 30% said they delayed buying a home or delayed their education. And 1 in 7, and if you do the math, that's about 15 million Americans, said that they had been turned away from or had to avoid the use of medical care because they were in medical debt. So this is still in our country a lurking problem. And I don't want to overemphasize it, but part of the reason that we need to talk about value and talk about pricing and cost is because as much as we want to celebrate new treatments, we have to provide them in a way that brings a balance to our system to address the needs of people who are struggling at the margins. And yes, if we're going to talk about equity, Black Americans are 50% more likely than white Americans to be in medical debt. And people who identify as Hispanic, 35% more likely. So this is a serious issue, and it's not equally spread across our country. And they're more than likely never going to be in the front of the room at the table when a lot of these issues are discussed. So we just need to keep them in mind. And just very schematically, we're here today because we're going to talk and focus, as we should, on the devastating unmet need for patients and families with ALS. This is where our focus should be. It doesn't mean that we shouldn't also remember that all of us are part of a broader community in which we have to come up with a way to have innovation and celebrate new things while creating an affordable platform that can work for all. So that's why we're here today, to deal with all of the tensions and trade-offs involved, and to try to make sure that we can address more honestly and transparently how we get to the right spot. So you've met the members of the Midwest Comparative Effectiveness Public Advisory Council. We convened them to help facilitate this public conversation and to give guidance on their interpretation of the evidence and to hear further testimony and consider the broader values involved. Let me give you a few words about ICER, which is the convening organization. As far as our potential conflicts, we keep our funding updated on our website. Our overall funding, as you can see, is dominated by funding from nonprofit foundations, the chief amongst which is the Laura and John Arnold Foundation, but we also have funding from the California Healthcare Foundation and several others. We do receive approximately 24% today direct contributions from life science companies and from health plans and provider groups. That is kept separate to help support our policy summit and non-report activities. And we are now currently also getting some revenue through our cloud-based platform of our results, the ICER Analytics platform, and very small amounts from government contracts and other philanthropy. The report that's the foundation for the discussion today starts about 8 months ago, and it starts with a very formal and intensive phase of what we call scoping. And that means that we go out initially to patients and patient groups, to clinical experts, to the manufacturers and to others. And we really ask them to orient us, to tell us what we don't know, to tell us what's important to them. Where are the potholes in thinking about the evidence? What's the arc of innovation? What's the unmet need? With that as guidance, we then start looking for evidence. We look at published evidence. We try to find if there's unpublished evidence that can be helpful in thinking more broadly about value. And we produce iterations of our report that go through public comment and revision. Towards the end, we were also glad and thankful to get expert reviewers, three of whom, at least two of whom, I'm sorry, are with us today, Drs. Bedlock and Shemaskin, and also Ken Menkhaus and John Turnbull were able to provide input into reviewing our report. Now, the evidence itself is kind of conceptually important to think about as we talk about value, because we do have a very kind of formal way that we talk and think about value. And it starts, if you will, in the green boxes with the things that are easier to measure. Health benefits. Does a treatment help us live longer? Health benefits. Does it help people return to better function? Does it have fewer side effects than other treatment options? Are there other aspects that can directly benefit their health? And then we do look at costs as part of value. And importantly, we're not just looking at the drug costs, we're looking at the impact of treatment on all the costs in the health system. And you'll also hear about a societal view in which we try to capture the impact on people's ability to work and other kinds of outside healthcare costs. We have those, the green boxes are kind of what's core to the cost effectiveness information that's in the report and that will be presented. And then very importantly, also in the report and part of our discussion today and votes will be discussions around the potential benefits of treatment that can be seen as lying beyond health, that arc into the family, that arc beyond year-to-year health benefits into a longer story of the impact on patients and caregivers. And we'll also talk about whether we have special priorities, commitments, ethical or otherwise, to consider the potential benefits of treatment differently given the context of the condition, whether there are other treatments available, its severity, et cetera. So all of this is addressed, and much of it is already informed by patient input throughout the 8 months, but it'll be an important part of our discussion today as we try to bring a broader view to value into thinking. So the agenda today will begin with discussion around the clinical evidence and the economic model evidence. Then these are all central time. We will break for lunch at 11:20, returning at noon for public comment from manufacturers and patients and others. Then we will have directly following that, we will be moving into a vote, deliberation phase from about 12:40 to 1:50. And then the policy roundtable at the end is where we'll have the patient advocates, experts, and clinical experts joined by representatives from the payer community and by manufacturers. And the goal will be to talk forward-looking, like, given the evidence, where are we today? What is this going to look like? What are the options for how insurers can cover it? What are some of the perspectives on good ways to provide insurance coverage, bad ways, if you will, to provide that? What do we think about pricing and payment options, and the future research that is always going to be part of our agenda at this stage of thinking about new treatments? So we anticipate those discussions culminating with final comments and adjournment by 4 p.m. Central. So with that, let me hand it over to Dr. McCann, our evidence author. Please introduce yourself, and then I'll let you begin the process with Reem moderating the discussion. Thanks.
Great. Thanks so much, Steve. My name is Anil. I'm the evidence author for this report. I'm an assistant professor of medicine at the University of California, San Francisco. I'm going to talk for about 20 minutes about the background of ALS, and we'll review the clinical evidence, and then we'll have about 25 minutes for question and answer period. I'd like to recognize several key collaborators, Rashid, Avi, and Dimitri, who are ICER staff during this report. I received funding from ICER as evidence author on this report, and I have no other conflicts of interest to disclose. So in terms of background of ALS, as most of you know, ALS is a rare, rapidly progressive and fatal disease that causes loss of motor neurons in the brain and spinal cord. The clinical presentation is highly variable depending on which motor neurons are affected. About 2/3 of cases are limb onset, where symptoms begin in the arms or legs, and about 1/3 are bulbar onset, which affects muscles of the face and neck and presents as difficulty chewing, speaking, or swallowing. The core feature of ALS is muscle weakness that often progresses to paralysis, respiratory failure, and ultimately death. The average life expectancy is only about 3 to 5 years after symptom onset. However, about 1 in 10 are considered slow progressors and survive over 10 years. In terms of the epidemiology, about 2 per 100,000 persons are newly diagnosed with ALS annually. There are approximately 25,000 people living with ALS in the US. The cause of ALS is mostly unknown, but it's thought to be a combination of genetic predisposition, environmental exposures, and aging-related dysfunction. And about 90% of cases are sporadic, meaning that they occur in the absence of a family history. In terms of risk factors, age is by far the strongest risk factor, with 60 to 79-year-olds having about a 17-fold higher incidence in developing ALS. Male, white race, and military service are additional risk factors for developing ALS. ALS is estimated to cost about $1 billion annually to society, but this likely underestimates costs from unpaid caregiving as well as loss of household income. Regarding the standard of care and management of ALS, the diagnosis is primarily clinical, and as such is often delayed about 1 year after symptom onset. The prognosis is worse for people with ALS if they're older, have bulbar onset, are fast progressors, and have decreased lung function. There's no cures for ALS, so the treatment is largely supportive care, which consists of symptom management, nutritional support to stabilize weight, and non-invasive ventilation to treat respiratory insufficiency. And these are ideally best delivered through specialized multi-center ALS centers. There have been over 80 published randomized trials on ALS, and Rilazole and Aderavone are the only 2 medications that are FDA approved to slow the progression of ALS. Both of these therapies are add-on therapies and are thought to be best when used upfront and can be used simultaneously at the discretion of the doctor and the clinician. Rilazole is an oral therapy and is thought to prolong survival by about 2 months in clinical trials. and is uniformly recommended by guidelines in the US and abroad. Intravenous Aderavone was approved in 2017 and is not endorsed by the American Association of Neurology in their reaffirmed guidelines in 2020 and is not approved for use in Europe. And of note, the oral formulation is part of our evaluation today. Several important insights from our discussions with patients and caregivers I wanted to share. First, that there's a diverse range of experiences and symptoms and progression. Caregiver needs and burden are profound, and this includes their time, stress, health, and financial. There's considerable enthusiasm for new treatments with a very high tolerance of risk. However, treatment burden and costs were cited as major barriers to trying a new therapy. For example, only some use intravenous Aderavone, citing limited evidence, risks from having a catheter, the burden of infusion therapy, as well as the cost of therapy. However, there's much more enthusiasm for trying oral drugs, as most use Rilazole, and many have expressed interest in trying oral Aderavone, even if they haven't used the intravenous formulation. There's two new medications to potentially treat ALS, which is the focus of our evaluation. The first is AMX0035, that I'll abbreviate as AMX, and is a oral combination of two existing drugs, sodium phenylbutyrate, abbreviated as PB, and taurursodiol, abbreviated as TURSO, or also known as TUDCA. The theoretical mechanism of action of this combination is that they target two different mechanisms of neuronal death. It can be taken daily for 3 weeks and then up to twice a day thereafter. Of note, the FDA Advisory Committee met on March 30th and voted 6-4 that the evidence did not support efficacy, but is reconvening on September 7th to consider additional analyses by the manufacturer, with a decision expected on September 29th. The second medication of focus is oral Aderavone, also known under the brand name of Radicava ORS, and it has the identical dosing schedule as the intravenous formulation. The theoretical mechanism of action is that it's a free radical scavenger, and it was FDA approved on May 12th of this year based on bioequivalence with the intravenous formulation. So regarding the scope of our review, I'm going to be discussing the clinical comparative effectiveness of adding AMX or oral Aderavone to the respective standards of care for adults with ALS. My colleague Kung Ho will be talking about the cost effectiveness in the next session after this. In terms of interventions, we'll be looking at both drugs against their respective standards of care without a head-to-head comparison. The standard of care for AMX is background therapy of Rilazole, plus or minus intravenous Aderavone, plus or minus specialty multidisciplinary care. And for oral Aderavone is Rilazole, plus or minus multidisciplinary care. And again, these drugs are both add-ons, and all of these drugs can be used upfront and at the same time. So now moving on to the clinical evidence. So before I talk about the clinical trials, it's really important to understand the primary outcome that was used in all the ALS trials that we'll be discussing. And this is the ALS Functional Rating Scale Revised, abbreviated ALS-FRS-R. So this scale consists of 12 items across 4 domains that you can see on the right. Each item is scored from 0 to 4, where higher is better, for a maximum of 48 points if you're completely functionally intact. There's no established minimal clinically important difference for this scale. However, a survey study of 65 ALS doctors found consensus that slowing of the disease by 20% or more was considered meaningful. Stakeholders that we spoke to expressed that a 1-point change is more modest, but can still be important. So I'm first going to talk about the clinical evidence for AMX, and then I'll turn to oral Aderavone. So for AMX, the evidence base is supported by the pivotal Centaur Trial and its companion open label extension. The Centaur Trial is a small 24-week phase 2 trial of early stage ALS, which included 137 participants who had a definite diagnosis of ALS, had early onset ALS with symptom onset of 18 months or less, and had intact respiratory function with a slow vital capacity of greater than 60%. Participants were randomized in a 2-to-1 fashion to the treatment arm versus the placebo arm from 25 centers in the Northeast ALS Consortium in the US. About 2/3 of the Centaur trial participants then enrolled into an open label extension where they received the study drug unblinded and were followed for up to 30 months. Notably, the long-term survival analysis included all 137 participants as originally randomized in an intention to treat analysis using a vital status sweep of publicly available records. So this slide shows the effect of AMX on the primary outcome of function, which again is the ALS functional rating scale. AMX had a 2.3-point difference in slowing of disease progression, which translated to about a 25% slowing of functional decline. This analysis notably excluded 2 early deaths in the treatment arm for people who received dose but died before undergoing an evaluation of function. So under the directive of the FDA, the manufacturer conducted a joint rank analysis where they combined function and survival and also found a statistically significant benefit for AMX with a p-value of 0.037. The FDA felt that other statistical analyses were more appropriate, so in their reanalysis of the data, they found lower efficacy and less statistical persuasiveness for both of these analyses, as shown below. So this slide talks about the effect of AMX on secondary outcomes. For secondary clinical outcomes, they were both not statistically significant, but numerically favored AMX. And this included the isometric muscle strength as measured by the Atlas measurement tool, and the slow vital capacity, or SVC, which is a measure of lung function. There's no known minimal clinically important difference for these scores, but a 5% difference in the slow vital capacity is considered clinically important. The Centaur trial also tested an exploratory plasma neurofilament biomarker, which was not statistically significant and actually went in the opposite direction as hypothesized and favored the placebo arm. Although it's hard to measure the neurofilament in plasma compared to spinal fluid, and experts that we spoke with doubted its validity as a surrogate biomarker. So what about the effect of AMX on survival? So this is the Kaplan-Meier survival plot where AMX is in the top curve in blue, placebo group is in the bottom curve in gray, survival probability is in the vertical axis, and time on the horizontal axis. And as you can see in the red circle, that there's no difference in survival during the first 6 months of the trial. And this was the randomized double-blinded phase. And that during the open label extension is where a treatment benefit emerged for survival with about a 5-month difference in median survival favoring AMX, which translated to a hazard ratio of 0.64 for death, which was statistically significant with a p-value of 0.048. In terms of the safety profile for AMX, there are minimal serious harms. Diarrhea and nausea were the most common side effects and were greatest in the first 2 weeks, occurring in about 33% of the treatment arm versus 20% of the placebo arm. And this in part led to more discontinuation due to adverse effects between treatment groups. There are 3 major uncertainties for AMX. First, do the findings generalize to more advanced ALS or minority populations, as the Centaur trial enrolled early stage ALS who are predominantly white? Second, are the findings from the Centaur trial that I just presented valid? And this is based on 4 reasons. First, there is a randomization implementation error where the first 17 patients in the Centaur trial were erroneously assigned to the treatment arm, and the following 9 patients were assigned to the placebo arm to keep a 2-to-1 randomization balance. However, sensitivity analyses that excluded these first 26 participants were reassuring. The second is concern for unblinding, because AMX has a bitter taste and can cause gastrointestinal side effects, as I just talked about, where 73% of the placebo arm who completed an exit interview were able to correctly guess their group assignment. The third, as I talked about before, was that the FDA reanalyses of the same data found lower efficacy and less statistical persuasiveness. And then lastly, the survival benefit was not seen during the 6-month randomized control trial phase, and was out of proportion to the modest functional gains on the ALS functional rating scale. To help answer these questions, there's an ongoing multi-center phase 3 trial called the Phoenix study, which is set to be completed in 2024. Then the last uncertainty is whether combination therapy of AMX is better than terso or tersodiol alone. Tersodiol is the cheaper of the two medications. It's currently available as a nutritional supplement. It's taken by some patients with ALS, and there's early pilot RCT data that suggests a similar benefit on the ALS functional rating scale seen in the Centaur trial. There's an ongoing multi-center RCT of terso monotherapy versus placebo being conducted in Italy, set to complete in 2023. So now for the evidence rating for AMX. So for all ALS patients compared to standard of care alone, adding on AMX received an evidence rating of C++, which means comparable or better. And what this means is that we had a higher certainty that AMX does not cause serious harm, but a more moderate certainty of a comparable to substantial net health benefit. So now switching gears to oral Aderavone. So the evidence base for oral Aderavone is supported by 3 small 24-week trials of the intravenous Aderavone formulation, which were conducted in Japan as part of the MCI 186 clinical trials program. And again, the evidence base for oral Aderavone is assumed a bioequivalence from the intravenous formulation. So the first trial for intravenous Aderavone was study 16, which was a negative phase 3 trial in a broader early stage ALS population. This was followed by study 18, which was a negative, even smaller trial, which was a phase 3 RCT in a more advanced ALS population. A post hoc analysis of study 16 found benefit in a subgroup of ALS patients with very narrow, well-defined early stage ALS, which comprised about 35% of the study 16 cohort. And then this finding was substantiated in the pivotal study 19 trial, which is the basis of our presentation and our evidence report. So this table summarizes the primary outcome of the ALS functional rating scale. And it shows the change from baseline in the ALS FRS score at week 24. And I showed study 16 and study 18, which were both considered negative, as reference. So for study 19, Aderavone slowed the decline in function by 2.5 points on the ALS FRS score, which was statistically significant with a p-value of 0.001. And this translates to about a 33% slowing of functional decline during the 6-month study. In terms of secondary outcomes, intravenous Aderavone improved quality of life with a decrease of about 9 points on the ALS assessment questionnaire, or ALSOC 40. And this was statistically significant with a p-value of 0.03. So the ALSOC 40 ranges from 40 to 100, where lower scores reflect better quality of life. Intravenous Aderavone also improved respiratory function numerically, but was not statistically significant, with a difference in the forced vital capacity of about 5%. Again, the minimal clinically important difference on this measure of lung function is unknown, but a difference of about 5% would be considered important. Grip and pinch strength were no different statistically or numerically between groups. There was no survival advantage for intravenous Aderavone. However, there are 0 deaths during study 19, and only 3 deaths emerged during its companion open label extension. So in the absence of robust clinical trial data on survival, we included a high-quality observational study recently published in JAMA Neurology, which found no survival difference from real-world evidence of 260 patients with ALS treated in 12 German ALS centers, as shown in the top left panel. From this population, about 104 closely approximated the study 19 criteria and is shown in the upper right panel. Controls for the study were selected based on being matched on the site of onset, being limb or bulbar onset, the age, duration of ALS, and the baseline ALS FRS score, which are thought to be 4 of the most important prognostic factors in ALS. terms of the safety profile for Aderavone, there are minimal serious harms in the randomized control trials compared to a placebo infusion. There were slightly more contusions noted in the treatment arm versus the placebo arm. Although not directly evaluated in any clinical trials, the oral formulation appears to be safer than the intravenous formulation, mostly just because of the nature of being oral and obviating the need for risky and burdensome catheter and IV infusion therapies. There are several uncertainties and controversies for oral Aderavone. First, as previously mentioned, 2 of the 3 trials of intravenous Aderavone were considered negative. Second, intravenous Aderavone was only studied in Japan, so there's uncertain generalizability to other populations and other health settings. Third, although both the intravenous and the oral Aderavone have FDA approval for all ALS patients, the study 19 criteria only represents up to 10% of all ALS patients. And then lastly, the evidence for oral Aderavone is assumed equivalent based on bioequivalent studies between the oral and intravenous formulations. So in terms of the evidence ratings for oral Aderavone, these were split by whether patients would meet the narrow study 19 criteria versus not meeting the narrow study 19 criteria. So compared to standard of care alone, for people who meet the narrow study 9 criteria, oral Aderavone received an evidence rating of C+, which is comparable or incremental, which means that we had a high certainty it does not cause serious harm, but only a moderate certainty of a comparable or a small net health benefit. And for people with ALS who do not meet the narrow study 9 criteria compared to standard of care alone, adding on oral Aderavone received an evidence rating of insufficient because we were not sure if it was effective in a broader population, and any harms, even if there was not a significant harm signal, could be net negative for people's health. So in addition to the clinical evidence that I just reviewed, there's potential other benefits and contextual considerations worth considering. First, the acuity of the need for treatment in ALS is extremely high. And I can't stress this enough, the caregiver burden is considerable and is likely underestimated in clinical studies. And then lastly, oral Aderavone overcomes the burden and risk of its IV formulation because you don't need a catheter and you don't need to come to clinic for infusion therapies. During the draft phase of our report, we received several public comments. For AMX, we received a request to include long-term tracheostomy, ventilation, free survival, and hospitalization data that was recently published from the Centaur open label extension. We agree that these outcomes are very important. We chose not to include these because there's considerable missing data for these outcomes because not everyone enrolled in the open label extension, and then a considerable number of people dropped out before the end of follow-up. A second comment that we received for AMX was a request to include recently published data on crossover adjusted survival benefit during the open label extension analysis. So we agree that the survival analyses that were presented are conservative because they don't fully account for the potential benefit of patients in the placebo arm who cross over to receive AMX at the beginning of the open label extension. However, we did not choose to include this because it's really difficult to capture the effects of crossover in an observational study based on the fact that there's several assumptions that are unknown for AMX and are not testable. For oral Aderavone, we received several public comments around the theme that the evidence supporting clinical effectiveness is robust and that we should consider upgrading the evidence rating. We agree that study 19 showed a modest improvement on the ALS FRS scale. However, our enthusiasm was tempered by the fact that only 1 of 3 trials were positive. There's less enthusiasm by clinical experts for using the medication. There's no change in survival, and that real-world evidence did not show benefit in survival or in function. So in summary, AMX appears safe and may have a modest benefit on disease progression and a more substantial benefit for survival, we had concerns about the trial conduct, analytic choices, and inconsistencies. For oral Aderavone, we thought it may slow progression modestly without a survival gain, but only for a narrowly defined early stage ALS population. We're concerned about it being a small single trial in a homogeneous population. However, oral Aderabone is safe and overcomes many of the burdens and risks of intravenous therapy. And with that, I will take questions.
Dr. Makam, thank you so much. That was an excellent, really well-organized overview. I'm going to open it for questions from the Midwest CEPAC, and I already see, Greg, you have a question. Please raise your hand if you have any questions. Greg, to you.
Yes, thank you very much. And thank you, Dr. McComb, for a very clear, very lucid presentation on this important subject. My question relates to the Centaur trial published in the New England Journal in 2020. And the primary endpoint, as you described, the ALS-FRS scale. I wanted to make the point that this scale, as you well know, contains 4 domains: a bulbar domain, a fine motor domain, a gross motor lower domain, and a breathing domain. Each of those domains is assigned 12 points among the 48 points in the scale. And this is not in the main paper in the New England Journal of Medicine, but in the supplement, the online supplement, figure S4 makes the point that only one of those 4 domains was statistically significantly favoring the active drug, and that was the fine motor domain. The other domains, bulbar, gross motor lower, and breathing, were null. So I just wanted to make this point. Again, it's not in the main paper, so if you just look at the main paper, you might miss it. But I think that it is another factor that needs to be considered in looking at the Centaur data. And I wonder if you might be able to comment further on this issue.
Great. Thanks so much for your comment, Dr. Kerfman. It's a really important point. I'm going to take even a step back. One step back is that the ALS-FRS score is a complicated scale because it includes many different items across many different domains, and they're not equally weighted. I mean, they are equally weighted, but for patients, it's a very heterogeneous disease where certain domains are more dominant. So it can be very difficult to interpret this at a patient-to-patient level. In terms of the 4 different domains, you're absolutely correct that the fine motor subscale was the only one that was quote unquote statistically significant and showed the largest benefit. But I would caution over-interpreting the subdomain scales just because it's a very small trial. So it's really hard for subgroup analyses, not just for the scales, but also by key characteristics. And all domains did show improvement, trended to show improvement for AMX compared to placebo. It just one was statistically significant.
Thank you, Dr. Markham. I see more hands. So Tim Wilt.
Thank you. Again, as usual, just outstanding presentation. So thanks a lot. I'm going to ask for some, I guess, interpretation or just comment on the C+ and the C++. But I'd like to walk it through a little bit, the presentation, because as presented, we had information about harms that were there, perhaps not serious, but more withdrawals in the first one. So there were harms, maybe not serious, and it resulted in more withdrawals. you then describe and actually summarize very well, particularly for the second one, why you had concerns about even effectiveness with 1 out of 2 studies maybe showing some benefit in the first drug. Sorry, you turn my camera on. Sorry. And in the first one, depending on the analyses, not statistically significant. And so as I read it, there is high certainty of no harms. I think there are harms that may not be serious, and moderate certainty of comparable or net benefit. But that would imply that there's at least comparable or greater benefit. And I think there's a lot of uncertainty that there's any benefit at all. That's very long-winded, but and I also want to support what Dr. Kerfman had to say about my concerns about the subscales. So I don't know if you can weave your way through an explanation, maybe through how you got to a C+ and a C++ given those. Thanks. Sorry.
Yeah, great. Thanks, Dr. Wilt, for the opportunity. I'll answer specifically about AMX, and then I may turn to Dr. Rind about the ratings in general. So in terms of AMX, you're right. There are harms with all drugs. There's always harms, and we do see an imbalance, especially in the gastrointestinal side effects. But these are self-limited. So we had a high certainty it did not cause serious harms. So thanks for the opportunity to clarify. And we did have, so there's certainly uncertain. It's a single trial. It's small, and as I discussed in the presentation, there are several unresolved certainties and controversies. But we did have a low to moderate certainty that the benefit on the ALS functional rating scale, if its precedence modest, and then there is potential for a more substantial benefit on survival that emerged in the open label extension, but granted with the caveats that I mentioned in the presentation, that it was out of proportion to the functional gains. It didn't emerge during the blinded randomized phase and only in the open label extension. However, it was assessed in all people as originally randomized and was a conservative analysis. I don't know, Dr. Rind, if you have thoughts about the rating scale in general, how to think about a C++.
I think it's important to realize that the comparator here is not giving the drug. So if we say that a C++ includes comparable, that includes that's a comparison to not giving the drug. So we have moderate certainty that one of the possibilities is it's the same as not giving the drug. Another possibility is a small benefit compared to not giving the drug. And another possibility is a substantial benefit compared to not giving the drug. To get to the substantial benefit with AMX, as Anil's talking about, that has to be that the survival benefit that we're seeing is real. The small benefit would be if that wasn't real, I suppose. But comparable includes no benefit. All of that is saying we're sure that there's no harm. Not no harm at all in that it doesn't ever upset your GI system, but that in a disease where we're talking about severities of rapid death, that the harms we're talking about aren't significant in thinking about the drug. And that was how we graded it. As you know, hopefully that makes sense across the two drugs. If people have more questions on that, I'm happy to try to explain it further. There's a graphic I think you all have in your evidence summary, I hope, that shows the graph of the evidence matrix and shows that it's a spread from no benefit up to substantial benefit is what a C++, that's the uncertainty that it's encompassing.
Thank you, Dr. Malcolm and Dr. Rind. I see more hands up, and I'm going to go in the order that they were raised. So Don Casey.
Reem, may I ask that Kathy Collett be allowed to jump the line and go in front of me? Would that be okay?
Absolutely. Appreciate that. Thank you.
Go ahead, Kathy.
Thank you, Don. You may live to regret that, but thank you very much. Just this, I'm a first timer here. Just a comment regarding the statistical persuasiveness of the AMX 035 at the FDA Advisory Committee meeting. I think it is worth noting that the statistical brouhaha was over joint rank analysis, and that Dr. James Berry, who literally wrote the book on joint rank analysis in ALS, said that the Amylyx use of the joint rank analysis in their statistical analysis was correct. And also there was one statistician on the AdCom, and he voted in favor. So he voted positively. So the statistician did not find the problem either. And just, these are just a couple of lived ALS experience things that I'd like to toss out to you. Survival isn't the end all when you're facing ALS as a family. And then the ALS FRSR misses a lot, and you just need to keep that in mind. So thank you very much.
Thank you, Kathy, for that insight and sharing it. I'm going to go back to the order, Don.
Is it okay if I respond, Rita?
Oh, absolutely. Yes.
Okay. Thanks so much, Kathy. I really appreciate your comments about the ALS FRS score. And it's really important to contextualize. We always focus on survival as the most important, but it's often not, especially with the disease that causes immense disability and it's rapidly progressive. Regarding your question or comment about the statistical persuasiveness, so I do want to say the reanalyses by the FDA still favored AMX. The one on the primary outcome, which was on the difference of the ALS FRS score at week 24, was a lower point estimate and was not significant. And one of the main differences between that and the primary analysis was the assumption of whether the ALS FRS score declines in a linear fashion versus a non-linear fashion. And that was the main difference in their approach. And then in the joint rank, it wasn't that the joint rank wasn't used appropriately. The issue, and I think part of the reason was how missing data was handled between both analyses. From the manufacturer analysis, it was assumed as last observation carry forward, which is statistical speak for saying that if we missed functional ratings later in time, we just assume that they stayed the same. Versus the FDA approach, which used a multiple imputation approach, which says that this is our best estimate of what the function would be, understanding that there's a decline based on their previous assessments. And I just wanted to clarify that point. Thank you so much.
I wanted to make one more point about the statistics, which is there was also a bit of a brouhaha at the FDA AdCom about whether it was reasonable to look at survival in the open label extension study and whether that was a pre-specified endpoint separate from a combination. And that came up a lot at the AdCom. To the extent we could tell, it did seem like a pre-specified endpoint. I know there were arguments about this. So we felt that first, it seems like an obvious thing to be looking at in the open label extension, but also it did seem to us that it had been pre-specified, although the FDA statistician clearly had concerns about that.
Thank you again, Anil and David, for the clarifications, and Kathy for making sure we discuss these important points. Don, back to your question.
Yeah, thanks. I first of all wanted to just comment quickly on what Dr. Greg Kerfman brought up relative to the domains. And I think I remember somewhere noting that actually the respiratory domain, I think is probably potentially the most severe in terms of influencing survival, but I don't know if that's statistically valid. I just remember seeing that. But two questions really. One is related to the fact that there seemed to be variable use of the ALS AQ40 and the functional status measure. This specific one, I know there are others, ALS FRR. I'm wondering, first of all, if you saw or are aware of any interaction. It seems like these two should go hand in glove, but I don't know that. I don't know if they're directionally congruent or not. And I think I raised the point because there may be some useful information in knowing that. I don't recall that any dissection of this was done, and I remember that our experts didn't seem to have much insight from the statistical standpoint. Certainly from a clinical standpoint, there was strong feeling. The other is really more of a comment because I think I know the answer to a question, but I brought this up to our group, and that is the notion of what multidisciplinary care is. And it seems as though in the context of a disease like ALS, where we know the trajectory, we just don't know the time of the trajectory, that the notion of advanced care planning and the components of that in sort of a package of interventions that is done early, since advanced care planning, we know from an evidence standpoint makes a difference early, had been consistently performed and standardized and presented in the context of even shared decision making, knowing that it's an ongoing process. So I'm sure that this was involved, but I'm not sure it was evaluated from the standpoint of how much was given and when it was given. And I think it's important to point that out from a generalizability standpoint, but I'd be interested in your thoughts about those two issues.
Great, thank you so much, Don. So the first question I just want to summarize and recap is the ALS FRS scale and the quality of life instrument, which is the LSAC 40, are they measuring the same thing? Are they collinear, so to speak? And it's hard to say, and maybe, I don't know if one of our clinical experts who might be using these instruments can speak to that, but it was also only assessed in the Aderavone trials and not in the AMX trial, which does not include a quality of life instrument as a secondary outcome. Dr. Bedlack or Dr. Lewis, is this familiarity with how these two scales?
So Dr. Bedlack, do you have an answer specifically about the correlation?
The correlation between the quality of life score and the functional score? Is that the question?
Yeah.
Yeah. So, I mean, it's encouraging to see that. It's one of the few trials that I've ever seen that had a positive quality of life score. I can tell you that the correlation is not as tight as you would expect it to be. There's something called the disability paradox that occurs in ALS and many other neurodegenerative diseases where quality of life is initially quite small, goes down quite a bit, but over time it can actually come up as people learn to kind of accept the disease and reprioritize their lives. But I did have my hand up for a different reason, if I can take a couple of minutes. Did you finish what you wanted to say, Anil?
I wanted to answer the second question first, if you don't mind, Dr. Bedlack.
Maybe I'll say something. We have about 2 minutes and I see 4 hands are up. I'm actually gonna take the questions from all 4 panelists currently, but please make your point short, as short as you can.
Second comment is it is a generalizability. All these trials are recruiting from specialized multidisciplinary ALS centers, but not everybody has access to them because they're not available in all geographic networks, as well as potential cost barriers.
Thank you. There's maybe a point if we can address later, but Dr. McBride?
Yeah, this will be quick, I think, and an excellent presentation. I just want to know about the slide 25, the survival curve. Does that include those deaths that were not in the study or not?
Yeah, so it does. These include all deaths for all people, randomized.
Okay, thank you.
Thank you so much. Dr. Shamaskin.
I would just re-emphasize the high patient tolerance that you pointed out early in the presentation. I realize it's all anecdotal, but in my experience, taking care of patients with ALS and in my own experience and of the people I've gotten to know with ALS, our tolerance for risk and side effects is extremely high, and I would encourage you to think about that when you do the rating of the two different drugs? Thanks.
Thank you so much, Dr. Shamaskin. This is very important insight. We appreciate you sharing it with us. The last question, Dr. Bidlock.
Thanks, Reem. Yeah, I just wanted to give this some context. I mean, I don't know if you all know how difficult it is to do ALS research. As was pointed out, there's a lot of different causes to this disease. There's tons of heterogeneity to patients. The outcome measures that we're straddled with right now are clinical measures that are noisy and slow to change. There's a tension between what is the purest science and what's best for humanity. In the purest science, we'd love to do 2-year trials where we keep everybody randomized to either the drug or the placebo for 2 years, but that's inhumane to take somebody with a disease that you know is going to disable and kill them prematurely and do that. We'd love to replicate studies multiple times to be sure that they're accurate. But again, think about what that means for humanity. We wait for that second AMX 0035 trial in 2 years, 50% of everybody with ALS today is going to be dead by the time we get those results. And the rest are going to be a lot more disabled than they probably needed to be. And the last point, Anil, is you didn't mention just recently in the Annals of Neurology, 2 of the world's best living ALS trialists, Dr. Chaikovitz and Dr. Scheffner, thoroughly debunked every single one of the FDA's criticisms. I mean, I think our community stands together in thinking that this trial of AMX 0035 is the best thing that we've seen in 100 years of ALS research. We've never seen a drug with this many positive outcomes. Thank you.
Thank you, Dr. Bedlock. I don't know, Anil, if you have any specific comments. I think we're all struggling with the fact that we have a very difficult disease. Options are limited. Unfortunately, and we're excited there are trials. It's just unfortunate that they have limitations, and it's to do our due diligence. We have to be explicit about discussing all that. Okay, well, thank you all.
So much.
Yeah, thanks. With that, I would like to introduce our next speaker, Dr. Gango Su, who is going to talk about the evidence and the economic modeling.
Yep, thank you. My name is Kang Ho Soo, and I'm an assistant professor at the School of Pharmacy at University of Pittsburgh. And as I was introduced, I will be presenting on the economic analysis of AMX and oral Aderavone for ALS. I would like to acknowledge and thank 2 key contributors, Dr. Josh Carlson from the University of Washington and Marina Richardson, who is a health economist at ICER. So the objective of the economic analysis was to estimate the lifetime cost effectiveness of adding AMX 0035 or oral Aderavone to their respective standards of care compared to standard of care alone for the treatment of ALS. So I'll be briefly talking about the methods. And here are the methods that we used in the model. These are very standard ICER procedures. I would like to note that we used a lifetime time horizon and we used a cycle length of 1 month. The primary outcomes that we looked at were cost per quality adjusted life year, cost per life year gained, and equal value of life years gained. And with regards to the EVLY, when a treatment is associated with an increased survival, the EVLY values that period of life extension of a healthy US adult. To simulate a patient's progression through ALS, we used the King staging system. And this King staging system is based off of the ALS FRSR survey that Anil had mentioned. So I won't go over the details, but in the first 3 stages of King staging, it's determined by how many CNS regions are involved. So for example, if a patient developed problems with handwriting, they would have arm involvement. And so if they previously had leg involvement as well, they would move from stage 1 to stage 2. This is what the King staging system looks like as a model that we used in the economic analysis. As you can see, patients can transition sequentially from 1 to 2 to 3 and so forth, or they could move non-sequentially as well from 1 to 4A or from 1 to 4B. And from any of the health states, patients could transition directly to death. So for this presentation, for this economic analysis, we modeled 2 separate ALS populations based on the available data and evidence, in line with the clinical evidence as well. So in the first comparison, we modeled AMX compared to standard of care alone, which included multidisciplinary care, Rilazole, and IV Aderobone. In the second analysis, we had a comparison of oral Aderavone to its standard of care, which included just multidisciplinary care and Rilazole. So we had several key model assumptions. We list 3 here. The first is that the relative treatment effect of progression of oral Aderavone was only applied to King stage 1 through stage 3, and we assumed that this treatment effect was constant through these 3 stages. Second, for oral Aderavone, we assumed that the proportion of patients who received treatment benefit among all patients was 35%, and this was in line with the clinical evidence that Anil presented, as we did not feel that patients outside of study 19 received treatment benefit. And lastly, based on the discontinuation data at 6 months from their respective pivotal trials, we applied a monthly discontinuation probability of about 3.5% for AMX and 0.23% for oral Aderavone throughout the patient's lifetime. Here are the key model inputs on efficacy and survival. So the treatment effect on disease progression and survival were really what drove the clinical aspect of the model. In line with the clinical evidence, we assumed that AMX had a separate treatment effect on mortality. However, we did not assume that oral Aderavone had a separate treatment effect on mortality. Here are the treatment costs that we used in our model. For AMX, we assumed that the annual treatment cost was $169,000 per year. And this was assumed based on the annual parity price to IV Aderavone from CMS's payment allowance limit. I also do want to note that the manufacturer of AMX, they recently submitted in their recent submission to the Canadian Agency for Drugs and Technologies in Health, the price that they submitted, if we used the current exchange rate from Canadian dollars to US dollars, was approximately $169,000 as well. For oral Aderavone, the annual cost that we used was $171,000, and this was based on the wholesale acquisition cost. These are the healthcare sector related costs that we used in our model. These costs were broken down into 2 components, recurring monthly costs and transitional costs. So the recurring monthly costs were costs, for example, for physician visits, for outpatient facility visits, and so forth. Transitional costs were one-time fixed costs that patients experienced as they progressed or advanced through the different stages. And as you can see on this slide, and as you'll see on the next slide, ALS is a very costly disease to treat and to manage, and the costs increase, as you would imagine, as patients progress. Here are the societal and indirect costs that we used. Again, these were broken down into recurring monthly costs and one-time fixed transitional costs. These are the utilities that we used, or quality of life measures that we used for our model, or the King stages. Please know that these utilities were derived from patients with ALS in a clinical trial in the United Kingdom. And as you can see, as patients progress through the different stages, quality of life drops down very low to 0.27. And we recognize that ALS is a horrible disease. I'll briefly go over the results. So first, the base case results for AMX. In the top table, we provide summary results. And so as you can see, when you compare AMX plus standard of care to standard of care alone, we can see that the drug costs and the total costs are much higher for AMX. However, with these increased costs, there is a gain in life years, quality adjusted life years, and EVLYs. If we look at the difference in the life years compared to the difference in qualities, we see that that difference decreases again because when we use qualities, it incorporates the low quality of life that patients experience in the later stages of the disease. When we look at the difference between qualities and EVLYs, we see that difference increase again because EVLYs values that extended period of life similar to that of a healthy US adult. In the bottom table, we can see the incremental cost effectiveness ratios for AMX compared to standard of care. And we can see that the cost per quality gain and cost per EVLY gain are very high. And especially for the cost per quality gain of about 2.14 million, this is much higher than commonly accepted thresholds. Here are the base case results for oral Aderavone. Again, compared to standard of care alone, we can see that treatment with oral Aderavone is associated with much higher drug costs and total costs. But with these increased costs, there is a marginal improvement in life years, qualities, and EVLYs. And in the bottom table, when we look at the incremental cost effectiveness ratios, we again see very high numbers of 11.98 million for cost per quality gained and 8.19 million for cost per EVLY gained. And again, specifically that 11.98 million per cost per quality gained is much higher than commonly accepted thresholds. Here we present sensitivity analyses for AMX. And these sensitivity analyses were to assess the uncertainty of inputs on model results. We can see that the most influential parameters were the clinical efficacy parameters on disease progression and survival. However, we can see that across all of the inputs, none of them really moved the needle in terms of AMX crossing over to the cost effectiveness range. And although we don't present the results for oral Aderavone, they were very similar to that of AMX, and they can be found in the final report. In the more robust probabilistic sensitivity analyses, none of the simulations that we ran were found to be cost effective for AMX or for oral Aderavone. So in certain situations when background health costs are very high, treatments that extend life may not be cost effective at any price. And so when this happens, it is ICER's policy to exclude all background costs in calculations to determine the health benefit price benchmark. And so that is what we did, and we will show the results of that on the next slide. So when we take out background healthcare costs to calculate the health benefit price benchmarks, you can see compared to the current annual cost for AMX of $169,000, the fair price or the health benefit price benchmark that we calculated was around $9,100 to $30,600. And these were based on various thresholds for cost per quality and cost per EVLY gained. And so for AMX, the discount needed from the current wholesale acquisition cost to reach the threshold prices was anywhere from 82% to 95%. For oral Aderavone, compared to the current annual WAC price of $171,000, the health benefit price benchmarks that we calculated needed to be around $1,400 to $3,200 annually. And so the discount needed to reach these threshold prices was anywhere from 98 to 99%. So we ran several scenario analyses in with our model. Of note, we ran a modified societal analysis, and we had similar results as the base case for both AMX 0035 and oral Aderavone. As I mentioned, we ran several other analyses, scenario analyses, and they're all detailed in the final report. And I'll also be talking about a couple of them shortly. So we do acknowledge that our model had several limitations. First, we used a placeholder price for AMX, as we do not know what the price will be in the United States. Second, we realized that we did have a lack of evidence on treatment effect heterogeneity. So we know that treatments work differently for different people. Some respond better than others. Also in ALS, we have slow progressors and fast progressors, and we don't know how the treatments would affect these subgroups differently. So based on the data that we did have, we just used an average treatment effect. We also know that there's probably a within King stage treatment effect for oral Aderavone and AMX, but we did not have data on this, so we used an average. So we used the treatment effect that we did have. We used it consistently across the King staging system. So we received several comments first on the appropriateness of only applying the Aderavone's treatment effect to 35% of the population, while we assigned 100% the cost of Aderavone to 100% of the patients. So we ran a scenario analysis where we gave 100% of the patients the treatment effect of Aderavone, and we found that it did not really change the results dramatically, and oral Aderavone was still not found to be cost effective. We also received an updated analysis on AMX's survival treatment effect. We thought that these the updated survival estimates were overly optimistic. However, we still ran a scenario analysis using these updated parameters, and we found that AMX was still not cost effective. And then we also received comments on using the King staging system versus other model schematics like the FT9. However, we do want to say that the King staging system has been used in previous economic analyses, and based on the data that we did have, the King staging system was the only model schematic that we could use. And finally, in a recent submission to CADIF by Amylyx, the manufacturer of AMX, we found very similar results that were in line with other model schematics. So in conclusion, AMX 0035 and oral Aderavone were found to provide clinical benefits in terms of gains in qualities, life years, and EVLYs over their respective standard of care alone. If priced similarly to the assumed placeholder price, currently AMX 0035 would not meet commonly cited cost effectiveness thresholds. And at its current price for oral Aderavone, it would not meet commonly cited cost effectiveness thresholds as well. Thank you.
Thank you very much, Dr. Su. Again, really very clear presentation for challenging topics. I am going to open it for questions, and I see Brad Martin, you have a question, please go ahead.
Hey, good morning. I had a question in the evidence reports, table 4.3. You also presented the slide, and this is the base case results for Aderavone. And my question revolves around the standard of care, and the drug costs that are reported in the results table work out to be $1,300. And my understanding that the standard of care arm also included Rilazole. And I don't know how, I guess don't understand how the drug cost in that comparison could be so low, given that Rilazole, and I did a real quick lookup, is about $800 a month, which would be about $10,000 a year, and with the life years of, we'd be way over $1,300. So I just wanted to ask and see if you could help my understanding on that point.
Sure. The price that we found was actually, so I don't know what the monthly price would be, but the price that we found for the wholesale acquisition cost, and we probably used the generic price, was 67 cents per tablet. Yeah, and so that's why when we costed that out, it ended up being about $1,300 per year.
That would explain it. Thank you.
Thank you. Let's see, Dr. Bora, you have a question?
Yeah, I have a question. I think Don asked briefly. So can you explain to me the standard of care cohort? It seems like, so for the first comparison, you have Rilazole plus minus and then IV Aderobone. So how do you actually tackle that? Because if it is plus, then you have to include the cost and everything, right? And if it is minus, I mean, how do you really include basically incorporate the variation with and without?
Sure. So for the treatment, so for the standard of care arm that you're talking about and when we used when we looked at the comparison for AMX, the percentage of patients that we assigned IV Aderobone to was based off of from the Centaur trial, which I believe was around 35% or so. So we applied we applied the cost to only a proportion of patients based off the standard of care arm that we found in the Centaur trial.
Okay, so you basically went with that proportion, whatever you found from the Centaur trial, and then okay.
And it may be related to that, and I see Dr. Bidlack hands is up, but just related to that, if you want to expand a little bit on the idea of a model without the background healthcare cost, is that something that's commonly done? Just educate us about, why did you make that decision, and is it what's commonly done in these type of analyses?
David, do you want to take that one?
I can, or Josh can, if you want to discuss.
Sure. So it's not common. These are costs to the system, but it is something to bring into consideration when you're considering fair pricing, because you don't want to penalize necessarily drugs that can't be cost effective for things that are already in the system and they're not necessarily cost effective already. So it is an analysis that was run to help sort of color the entire discussion, but it's not standard outside of that specific context.
Thank you very much.
I can go slightly further than that if you want, Reem, which is, just imagine that a drug like AMX, if we believe the results, extends life for 5 months. You don't want to say we're not willing to pay for this drug because extending the life of somebody with ALS is too expensive because the costs are already not cost effective. And so when ICER bumps into a situation like that, we're not willing to penalize a new life extending drug for the fact that the background costs are too high already. We want people to get new life extending drugs, even if the background costs are too high.
Thank you, David. That was extremely helpful. Thank you. Okay, Dr. Bidlack.
Hey, Dr. Sue, I'm not a cost effectiveness expert. You mentioned this term commonly accepted cost effectiveness standards and that neither of these two new drugs meet those, but how can there possibly be one cost effectiveness standard irrespective of the severity of the background disease and the unmet need? I mean, surely society recognizes that a new blood pressure medicine or migraine medicine, where we already have so many other options, should have a different standard than a new ALS medicine, one of the world's worst diseases for which we really have no effective therapies.
Yeah, so I would agree that ALS is again, it is a horrible disease. But it's the reason in this case specifically too why the cost effectiveness ratios jump so high is because again, so when we have a small incremental gain in life years and qualities, but a big price difference between the active drug and the comparator, we're going to get big cost effectiveness ratios. Yeah.
So I can help a little bit. There are common standards that need to apply across all treatments because of the trade-offs that Steve mentioned earlier. And that's basically a level set so that we can make decisions across the entire healthcare system. There is the potential to bring in other considerations in a deliberative process like this, and this process does allow for those other considerations, but you start with a standard approach that applies this sort of standard to all interventions across all diseases so that we can appreciate those trade-offs. And again, then you bring in the contextual considerations for a specific disease when you're making specific decisions about an intervention of interest.
Thank you. Okay, Dr. Shemaskin.
Yes, I have two questions and then one comment. The first question relates to the EQ5D questionnaire that I believe was used in your analysis. Those questions, it seems, really aren't customized to fit with the life of a person with ALS. These questions don't appear to be really relevant. And my question is, how can you use the data from that in your analysis?
Sure. No, thank you for making that comment and for the question. So the EQ5D, when you measure a patient's utility, it is a validated measure. But so we did receive comments that maybe these are not the right utility measures. But if you look at the sensitivity analyses that we had, especially for the tornado diagram, and as well for the probabilistic sensitivity analyses, the utilities really did not really move the needle at all in terms of concluding that AMX or Aderavone were not found to be cost effective.
And I'll just add that these scores were taken from patients with ALS. And so that's important to consider. We also did request additional evidence from the manufacturers, from their trials. There are different ways of calculating utilities. These data were not provided to us. And so these are the utilities that were available and have been used in various analyses and are in the public domain.
Okay, thank you. My second question relates to the overall uncertainty of your cost assumptions. My understanding is, and you did emphasize this, but my understanding is a lot of the data came from other patients in other countries and studies in other countries. And given the not just diverse population of people with ALS in this country, but the broad and different healthcare system here, how confident do you feel that those results are really generalizable to cost assumptions here? And then wouldn't that affect your total cost effectiveness analysis?
So yes, thank you, Dr. Shemaskin, for that question. And so you're right. Some of these, some of the costs were taken from other countries because that was the best available evidence that we did have. We did, as Dr. Carlson mentioned, we did ask for these types of data as well from companies, but we did not receive them. So if we knew the quote unquote true cost, yes, the total cost would probably change by a little bit, but the incremental cost when we look at the active drug compared to a comparator would probably not change too much. And we can determine that based on, again, on the sensitivity analysis that we did, where we varied those costs, and we did not see that even when we varied the cost one by one or together simultaneously, it did not really change the conclusion that we came to that AMX or Aderavone were cost effective.
Correct me if I'm wrong, but wouldn't CMS have that data for patients in this country on IV Aderavone already?
So I'm sure they could conduct a study to find the cost to these patients by stage, but based on the published available evidence that we looked for in the public domain, we did not find this information.
So my comment relates to when I think about the voting questions you're going to be addressing later. I see a historical parallel between the status of ALS treatment now and what was in existence in the early 1970s and mid 80s related to breast cancer and AIDS. In 1974, Aderomycin was introduced, and there were lots of concerns and uncertainty and definite cost issues. And then in the mid 80s, when AZT was introduced, again, there was a lot of concern that it might only help certain subgroups of patients, and cost and side effects were also an issue. I can speak from both professional and personal experience that although the diseases are different, the principle is still there that when you give patients with, or as we did, patients with those diseases, hope and something real to use, the concerns about cost become diminished. And I think that if we could go in a time machine and go back, if a cost effectiveness analysis like this was done then, I think we'd have to wonder if the approval of those drugs would really have moved along as quickly as they did, and if society would have been helped as much as they were. Thank you.
Joel, I'd be interested in answering that because as you probably know, the first AIDS patient I ever saw was at your institution when I was a medical student there. And I can remember when the drugs for HIV came out, I really did a ton of AIDS care early in my career. And the drugs came in at about $5,000 a year, one after another. And so it cost about $15,000 a year. And there were tremendous concerns about the price. And I had done a little bit of cost effectiveness work back in those days, and I thought people were insane. Here were these drugs, not that extended life by 4.6 months, but that took young adults who were on hospice and saved their lives and brought them back to work and kept them alive for decades. And people were worrying about spending $15,000 a year. And that didn't even count the cost offsets of not having them end up in the hospital. And everyone who did a cost effectiveness analysis of drugs for HIV thought that the complaints about the pricing were crazy, that every cost effectiveness analysis showed that those drugs were incredibly cost effective because they were so good. So I'd be cautious about thinking that a cost effectiveness analysis would have led to pushback against the AIDS drugs that you and I were all so happy to get and so happy to administer. They were spectacularly great drugs and really cost effective. And the cost effectiveness was because they worked.
And I'm going to jump in just because I'm selfish and I want to save some of this good conversation for the policy roundtable. It's a great perspective that we want to wrestle with in a variety of different ways for sure. Handing it back over to you, Reem.
I agree. But really, really very important points, and kind of thinking about where do we draw thresholds for decision making. And I just want to, although very hard, but we're going to try as much as we can to think about, you know, the effectiveness separately and then cost maybe later, and try and make judgments around these. I see Dr. Lewis' hand is up. Please go ahead.
Well, thank you. I mean, I was just going to just continue what Dr. Shemaskin said, because closer to home, historically, Rilazole was a drug that came out in the early 1990s, and at that time there was nothing for ALS at all. And the study was a small trial published in the New England Journal. It was only about 120 patients or so, and it showed a survival, but it was small. But what a whirlwind it was for ALS patients that have something to hold on to at that time. And there was the same discussions about cost effectiveness and the price of the drug that was very big. Now we are, 30 years later, and we're dealing with some similar discussions with similar results in ALS again. And so I think it's just historically, helpful to look back and see where we've come with ALS as well, and what that brought to the ALS community when there was first one drug finally available.
Thank you. Again, I'm sure Steve is taking notes because he's going to bring some of this back up in the policy roundtable. Eric.
Hi, Maureen. Thanks. I have a question about the standard of care cost, and we kind of describe this as a high cost condition, but experience tells me that the cost distribution for a patient with ALS is not symmetrical, meaning that the 4.8 months or 5 months of additional life that might be gained from a treatment like this may occur in a low cost period where you have higher quality of life during the trajectory of decline, because everyone is on a trajectory of decline. And so when I think about this, I mean, have we appropriately priced, sort of costed out the delay progression period with a cost structure that represents what the cost structure would be during the period of time where we're getting the benefit? So I mean, it's a nuanced question, but I'm curious if how people think about what I believe to be an asymmetrical distribution of the cost over the progression.
Yes, thank you, Dr. Armbrecht. So you may be absolutely right in that the survival benefit happens earlier for some and later for others, and just given the data availability, we didn't have that data, and so we had to model what would happen for the average patient. And so when we modeled, so the results that you see are for the average patient, and we just don't have the data right now, and we did not receive any additional data from manufacturers to kind of address your questions better.
And one aspect is, so that the effect on progression and mortality would have impacts on each of the stages as patients progress through.
But
so you're going to get.
But would it? I mean, does it? For
Yes.
example,
it's consistent. The treatment effect is consistently applied to each stage. Now, it's based on a natural history, on a trajectory. So the amount of time patients spend in any one is based on standard of care, but then the treatment benefit applies to each of those stages because it's consistently applied to each time in each stage. So you will have additional time in each stage.
Yep. So that's how the model. That's how the
model works. Correct.
Yes, that's how the model. I'm saying the question is like, is that really what happens, right? That is to say, does it equally distribute an extra month of a late stage, highly palliative, highly disabled period at the end of the progression? Or is that the same amount for every patient? And the benefit is actually in the lower disability, higher functioning, period of the condition, in which case we would all acknowledge like the cost structure of that period is lower, or at least I would say that it's lower.
Yeah, that's an empirical question. I don't know that we have the data to answer that, but it's certainly a reasonable question to ask.
Okay, thanks.
Okay, thank you. And I see Kathy, your hand's up. Please go ahead.
Thank you very much. And this is just a really quick observation, a trip down memory lane for me. My mom was diagnosed in late 1996. Rilazole, the brand name product, was new. She wanted to try it. Cost-wise, my parents were not wealthy. And cost-wise, it was doable with or without coverage. She said, we save for a rainy day, it's raining if insurance won't cover it. So it was that kind of a cost. I believe that if this happened today, I don't know that she could have accepted that the cost in these 6-digit numbers. So just a change in the cost perspective since 25 years ago and today. Thank you.
Thank you very much, Kathy. This is kind of very helpful from a caregiver and family perspective to remind us and anchor us around realities. Very, very helpful. Thank you. Okay, I actually am not seeing any other hands up, but I know, Stacy, you've had a question and you kindly lowered your hand earlier because of time. And I would ask you to maybe state it's a comment more now because some people have responded, but I think that would be helpful just to make sure we all hear it.
Sure, thanks, Reem. And this was on the clinical evidence presentation, and I had just had a question about the guideline recommendations around the IV Aderavone drug, and whether or not the recommendations, it was stated that that drug was not guideline endorsed. And I just wondered if there was any kind of statement about the oral drug potentially being more supported because it does lower the burden on patients and caregivers, or if any of that was really had any nuance around the patient population who might be represented from the trial, a subgroup of patients who may benefit.
Thank you, Stacy. And maybe Dr. Lewis, if you want to respond to that.
Sure. There's been no guidance from the American Academy of Neurology updating this as of yet. I think among the ALS community and physicians that take care of patients with ALS, certainly having the oral option is huge for patients to deburden them from the IV administration. It is a problem for many patients to get access to the drug through insurance because we have to submit authorization requests, and there are, it is being covered mostly only for those patients who fit the strict criteria from the trial, such that the patients must be early with very little respiratory involvement and early and only 18 months or 2 years based on which drug is being offered, or for just Aderavone. So it's hard for patients to get it, and the authorization is burdensome. And for those that don't meet the strict criteria, they're left out in the cold.
Thank you, Dr. Lewis. And again, really excellent points that I'm sure we'll discuss further in the policy roundtable. Again, I don't see any hands up. So I think with that, Steve, I'm going to hand it to you, and we probably can go to our break.
Thank you, Reem, and thank you, everybody, for a very, very good discussion, to which we will return after some lunch. So we're going to break now and reconvene at noon in Central Time, 1 p.m. Eastern Time. And at that point, we will have public comment followed by further deliberation and voting. So thank you, and we'll see you back at noon Central Time.